Hi everyone, welcome.
Hello.
Here we go again, Rihanna.
Yes.
I was missing you, dude.
Yes. Yeah, it's we didn't do so many, so it's a long time we didn't do a workshop together. So
Yeah, this season was a
like
little bit
all
different.
time.
Yes. Yeah, welcome back. I'm Felipe Matias. I'm here at North Dakota State University.
And I'm Anaita Marrano. I'm from University of Massachusetts, Boston.
Great. And today we have another workshop from our season. Is the 9th workshop. And as traditionally, we start with our introduction.
Yeah, so in this introduction, we usually explain who we are and what we do. So we are PhenomeForce, and we are a platform that aim to bring together people working in phenomics. So our aim is to provide resources to people in any kind of form. We started by organizing this workshop, this free workshop online that also try to empower early career researchers. As you will see today, we have give priority to PhD students or postdoc presenting their work and their application. So we have been trying to connect people in different way. First of all, we have a website where that we keep updating with initiative and events, so you can follow us there. We also have a Twitter account, so where we usually put like advertise our events, so you can follow us there. We have a YouTube channel where you are actually seeing us now, so we suggest you to subscribe so you will get notification. And also here you can see that we have all our repertoire of our repository of videos and workshop that you can always go and watch it and watch them again. You can also contact us by email. Our email is phenomeforce@gmail.com, and you can also send us an email if you want to join us on Slack. Slack is a workplace. It's an application that helps people to connect. So if you want to be part of this, we can add you. You can join any channel that we created based on the topic. And also, like we suggest you, once you join, to introduce yourself in the introduction channel. So people know what you do. If they want to collaborate with you, or if they have any question, they can contact you privately or in the channel itself. So also, if you have any suggestion about the future workshop or this workshop, or also if you have any tool or open source resource that you want to share and you want to present, please contact us. This can be useful also for the future workshop that we are going to organize. And yeah, so as I say, this is the YouTube page, our page, and you see that we have many workshop, many are also to come. Please subscribe so you can, as I say, get notification. And so we started last year with the first Friday hands-on workshop series, and based on the success that we get, we continued and we created this new one that Philip is going to talk more about it, right?
Yes, let's do it. So sign up here and follow us in YouTube. Also give the like and share these videos with your community, because our main idea here is sharing and learn with each other. So we need your help and to make this platform work and help more people. This season we are working, it's our second season, as Anna said, and it's focused on setting up low-cost phenotyping platforms. And we start this season on April 9th, and next week is going to be our last workshop from the second season. In the main focus here, when we talk about setting up low-cost phenotypes, is making the platform per se, but also collecting data on the cloud, or finding valuable data there to allow people to do research and learn together, right? So in this season, we are in partnership with other colleagues that is not here today, which is Claire from Agriculture and Agri-Food Canada, and also Noah Falgren from Dunford Plant Science Center. Today, the speaker give us some links. They are all here in the description, but also we send for in advance for if you register for this workshop. So that's what also is important, register for the workshops, because Anna and I and the PhenomeForce can have an idea about what to do next, and also idea who is following this workshop series. And with this, I want to introduce our next speaker today. We are really happy to have a Jenny here. Jennifer, but we are calling Jenny. And she's going to introduce more about her work, what she's doing, and she's going to talk about use of Arduino platform for obtaining long-term PhD reading in plant tissues. Hi, Jenny.
Hi, Jenny. Welcome.
Thank you so much for having me. I'm so happy to be here. So I'm a second-year master's student at Mayo University. I'm working in a pulse breeding and genetics lab under the supervision of Dr. Valerio Oyas Viegas. So I'm going to share my screen, and then I can begin my presentation.
Awesome.
So
yeah, and again, guys, we are using internet, many computer technology. If one of us lost the connection, just stay there. We're gonna do our best to come back in time.
Okay. Also, like as I say in the chat, if you have any questions, you can write them on the chat, and then we will ask Jenny to answer. So good luck, Jenny.
Thank you.
And
have fun. Yes, we'll be right here.
Alrighty. Sounds good. Okay. So I've shared it. Okay.
Did you share your?
I did, yes. I see it here, so.
Okay. Here we go.
There it is. So I'm going to full screen that so everyone can see. Okay, all right.
Bye-bye.
So thank you everyone for being here, and I'm super happy to be telling you all about my project, the use of an Arduino platform for obtaining long-term pH readings in plant tissue. So I wanted to start off by talking a little bit about plants and pH. It's not something that a lot of us really think about, but pH is really important for plant health. In general, the normal pH within a plant ranges from 6.5 to 8. This, of course, can vary depending on the cellular compartment, as well as it can vary between species. However, in general, any deviations from this normal pH can greatly impact cell function. So external pH can influence the internal pH. And where I'm getting with this is that certain plant pathogens can actually alter the pH of their host and cause infection. So let's dive a little bit deeper at what's happening at the cellular level. So I've drawn up this image here, and it's showing a fungal pathogen that is invading a plant. And so the fungus will produce these structures called hyphae, and that will penetrate the surface of the plant tissue. Going a little bit closer, let me see if I should get a pointer going. If we look more closely at what's going on, there's actually the production of organic acids, and this can include oxalic acid or citric acids. What this does is it actually lowers the pH of the plant tissue, and this creates an optimal environment for the function of any cell wall degrading enzymes that the fungus produces. And so these cell wall degrading enzymes can then go in and break down the cell wall of the plant cells and, of course, allow for the hyphae to penetrate deeper into the plant cells and propagate infection. So my work is mainly focused on white mold in dry beans. White mold is caused by Sclerotinia sclerotirum. I'm just going to refer to it as Sclerotinia. And it's a fungal pathogen that produces high quantities of oxalic acid. So this will decrease the pH of the plant tissue from approximately 6 to 4, enhancing the function of numerous cell wall degrading enzymes that it produces and promoting pathogenesis. So white mold is a devastating disease. It's very problematic in temperate climates with high humidity, and it also has a very wide host range. It can affect over 400 different plant species. The crop losses that result from white mold can range from 0 to 100%. It is quite variable, but there's always a possibility that you get complete loss of your crops. And so when it comes to dry beans specifically, there was a study that was done in North Dakota, and they found that the losses, the economic losses resulting from dry beans can be almost $3 million on average each year, and that was over a 3-year period. So this disease can be quite detrimental. So one of the telltale signs of white mold is these stems that appear to be bleached. So they look really pale, they come off as white or ashy gray, and it's very, very distinct. Another symptom is the wilting of leaves, and of course the appearance of this cottony white mycelium. So you can quite easily spot these symptoms. As the disease progresses, you will get the development of sclerotia. So sclerotia are these blackened hard masses, and they act as survival structures for the white mold. And the sclerotia can actually persist in the soil for a number of years, so that when the conditions are optimal, it can begin the infection process. All right. So when it comes to actually measuring pH, I've summarized here a table of the current methods that have been used in the literature, and I'm going to go through each one. The first one is pH indicator in agar. The indicator will essentially change color based on the pH, and this method has previously been used to measure the pH of root surfaces. However, to my knowledge, I don't think it has been used in leaves before. It is a fairly inexpensive method, so it is beneficial in that sense. The second method is the pH of apoplastic fluid. And so fluids are essentially extracted from using either centrifugation, vacuum filtration, or through a pressure chamber. From there, the pH can then be determined using a pH meter. The issue with this method is that it's very destructive. As you can imagine, the fluids are essentially being drained from the leaf, so you will not be able to measure pH on intact plant tissue. The third method is weak acid influx. So this is a method that allows you to indirectly infer the pH by looking at weak acid uptake in plants. So this is based on the concept that weak acids can easily move across the membrane. And so if the cell is acidic, you can infer the pH depending on how much weak acid is being taken up into the cell. What's nice about this method is that it is non-invasive, but the problem is it's not very well validated, and various external factors can actually influence the uptake of weak acids. So proton selective electrodes are a really popular method. For this, microelectrodes can be inserted directly into the cytoplasm, vacuole, or stomata of plant cells. Alternatively, these microelectrodes can be placed in a solution that's been equilibrated with plant tissue, and it allows for direct measurement of pH. The problem with these is that the microelectrodes are very difficult and time-consuming to produce. As well, you need a lot of experience to be able to know how to use them. And the last method I wanted to mention was fluorescence or confocal microscopy. I've lumped these two together because when it comes to pH measurements, the only real difference I could find between the two is that confocal microscopy offers slightly better resolution than fluorescence. But both of these rely on pH-sensitive fluorescent probes, so either as fluorescent dye or fluorescent protein. And so how this works is it basically measures the pH of intact tissue by exciting the probe using a specific wavelength. And then it will measure the emission of two different wavelengths. And one of them is going to change depending on the pH, while the other one remains the same. So you can directly measure the pH. The issues with this is that if you are using dyes, unfortunately, the fluorescence doesn't last very long. But if you are going to be using proteins, as in transgenic plants, then not all plants are necessarily suitable for transformation. So that could be a problem. Another thing is that the pH range of the response is limited, and of course you will need access to a fluorescent or confocal microscope in order to do this. So in our lab, we decided to ask ourselves, well, can we come up with a method that is fast, reliable, non-invasive, and cheap? And so we decided to look into pH sensors. These are miniaturized sensors that have been developed by the Bendable Electronics and Sensing Technologies, or BEST group, from the University of Glasgow. And they were very generous in providing us with a number of different sensors for us to test out. So I'm going to talk a little bit about the different components of these sensors. So first of all, you have a polymer substrate base on which the sensor is printed. There is a reference electrode, and in this example, it's made out of carbon, and a sensitive electrode made out of graphite polyurethane. So how it works is on the surface of the polyurethane, once it comes into contact with a pH solution, an electrical double layer is actually formed, and this generates an electric potential. And this electric potential can be measured as a voltage reading. And we decided to do that by using an Arduino. So Arduinos come in a number of different models, and each has their own specifications. The one we decided to go with was the Arduino Uno Rev 3. This model is extremely affordable, and it allows us to perform what we need in order to carry out our experiments. So I'm going to talk a little bit about what the Arduino consists of. So first, you have this USB connector here. And this allows the Arduino to be connected to your computer through USB. And all you really need is just a basic printer cable to do this. We also have a port for power, and it uses a DC barrel plug. You can also power your Arduino by using the USB connector. It's up to you whether you want to do that or not. And on the Arduino, there's also a number of pins. So we can see here that there are digital pins at the top, ranging from 0 to 13. At the bottom here, we have some analog pins going from A0 to A5. We have ground pins. There's a ground pin here too. And then there's a 5-volt pin here. And so these pins are the ones that I will be using in the demo today. So now that we know that the Arduino can be used to collect voltage reading data, how are we going to be able to store this data? And so we decided to use a micro SD card adapter that's specific to the Arduino. It has 6 different pins, and on the left side here, I've outlined exactly which pins they need to be connected to on the Arduino itself. I will be referring back to the slides once I do the demo for you guys. Okay, So this here is the schematic of what the setup looks like. I'll be using a breadboard. Breadboards are really convenient when it comes to any kind of circuitry. And how it works is that it's split down the middle. So the left side and the right side, these are not connected. But each row of 5 pins, these would all be connected. So each row is its own little connection. On the sides of the breadboard, you also have the positive and negative strips, so the positive going down and the negative on the right here. And the positive is connected and the negative is connected. Okay. So here are 2 images of my setup. The overall thing just looks like that, and then that's the sensor, and it's connected to a leaf. I've just connected it using some vinyl paper clips, and these are not going to conduct any electricity, so it won't interfere with any of the circuitry. Here I have a list of all the materials I use. As you can see, the most expensive item on this list is really the Arduino, which we all know is super affordable. So it's really, really inexpensive to set up. The total cost without the sensor is $87 in Canadian dollars. I do want to point out that this setup here allows for up to 6 pH sensors to be attached. But for simplicity's sake and for the demo today, I'm only going to be attaching 1 sensor. Okay, so now I'm going to talk a little bit about the code. So the code is super important because it basically tells the Arduino what it's supposed to be doing. And the program I like to use is the Arduino web editor. It's free. You just need to make an account. Everything's online. You can access it from a number of different devices. So this is the platform that I prefer to use. If that's not your style, you have the option to download the Arduino software. Again, it's free, and they offer it for a number of different operating systems, as you can see on the right here. So really, it's up to you which one you want to use. I personally prefer the web editor just because it's very easy to use. Okay, so now we have the code. So if you go to the PhenomeForce website, I believe you can access the link here. Everything is just on the GitHub. And so I'm going to just give everybody a minute to kind of just access this website here. And if you're having difficulties, please let me know because I do want you guys to follow along with the code. Okay, so let me see. Okay. Oh, does anybody have, I think, should we wait until everybody is able to access the website?
Yeah, I guess we can continue going. The link is right here in the description.
Yeah, okay. So if everyone's there, I'm going to switch over to the editor. So, okay. Okay, so I've switched over to the editor, and I think everyone should be able to see that now. I'm going to zoom in on the code so you guys can see. So here is the entire code, and I'm going to just mention some key points. So the code up here is really just to initialize the Arduino and have it set up for what we want to do. I'm not going to mention too much about it. There are 2 portions of the code I think that are important, and that's the setup, where the code is only run once, and then the loop, which is run over and over and over again. So that's down here. So within the setup, I have it so that it will tell me when the SD card is ready to use. So the SD card, again, is just going to be where the data is being collected onto. And in the event that something goes wrong with the SD card, I have it so that it tells me, and it will say the initialization has failed. And I only need to know once because I don't need it to tell me over and over again. All right, so onto the actual loop. This section of the code is where the voltage reading actually takes place. So the voltage reading comes from the analog pin, but analog needs to be converted to digital, and so that's what this line of code does here. I also have the time converted into an easier to read format in hours, minutes, and seconds. This part of the code here is really optional. It's up to you. I like to have it because it will tell me if there's anything wrong with my readings, if there's something I need to change. And basically it just prints everything to a serial monitor, and I'll be able to show you guys that later. And lastly, we have the code that writes to the actual SD card. And so it will create a file, and then it will write each reading as a line in the file. In the event that something goes wrong with the file, it's not able to open it, or it can't write to the file, then it will just say error opening. I also have it so that it's taking readings every second, and this is optional for you guys if you want to take reading every 5 seconds, every 10 seconds, really it's up to you guys. So I have a delay as well. So that's it for the code. I think we can now go over to the actual demo where I set everything up for you guys and show you exactly step by step from the beginning how to set this all up. So if you guys can help me switch over to my other device, and then I'm going to mute myself here and
You're gonna use your presentation?
Yeah, I mean, I think she's going to share the Chrome page.
Okay.
Very interesting after many workshops on Raspberry Pi to have another tool, right?
Yes.
yeah. Okay, so I'm ready to begin now. You guys can hear me, audio is good?
Yes, we can hear you.
Perfect, okay. And I think if you guys want to interrupt me for this, if somebody has any questions about how I'm setting everything up, then please let me know, and then I'll stop and answer any questions you have.
Okay.
Cool, thank you.
All right, no worries. So let's begin. So as you can see, here are all of the components that we need to set it up. I'm just going to run back to my slide because I need to refer to something. Okay. Okay, so here is basically the SD card that I'm going to be using, and this is the first thing that I'm going to be setting up. Okay, so on the back of the SD card, we can see that there are the 6 pins, and based on what I had in my slides, I'm going to just use my jumper wires to connect the pins to the breadboard and then to the Arduino. Where you place the SD card adapter is really up to you. I'm just going to stick it right near the bottom. And so it's fairly easy. Once it's stuck in place, it should not move at all, and you can see that it's not moving. So next I'm going to connect the wires, and I like to color code them because it makes it a lot easier to tell which wire goes where, and it's just really personal preference. But I'm going to use the exact color coding I did in that one slide I have in the presentation. So the CS pin should go right over here. So CS is blue. I've got a yellow pin here, or yellow jumper wire, sorry, that goes to the SCK. You can see that. My green wire is going to connect to the MOSI, like so. All right. Next I have a lovely brown jumper wire that I'm going to connect to the MISO. I also have the VCC, which is purple. Okay. And last but not least, we have our ground. So I'm going to just connect that in there. Okay. Okay, So everything has been connected nicely, and you can see that there. So now that I'm done connecting everything to the breadboard and these are now linked, I'm going to attach the jumper wires to the Arduino. And I'm going to be using the digital pins on the sides. You can see what the pins are. So my blue pin connects to 10. So I'm going to do that. My yellow pin connects to number 13. Green goes to number 11. Right, like that. We've got brown connecting to number 12. Okay. Okay, I'm just going to twist 13 because it's... Okay, there we go. All right, nice and connected. Okay, purple, we're going to go to pin 5 volts, so that's in the back, that's there. Oh, if you guys can see that. Oops. Okay. And we also have the gray, which goes to the ground. Okay. So everything now should be nice and attached, as you can see here. I'm going to bring the camera up close so that you can have a better look at how everything has been attached. So like so, and like so. Okay. So that's part's been set up, and that's really just for the data logging. And then part would be the actual sensor. So I'm now going to connect the sensor. I have a red wire and a black wire. The red wire I'm going to connect to the analog 0 pin, and then the black wire I'm going to connect it to the ground. So analog 0 and ground. So I'm going to zoom in so you can see there that you got the analog 0 there, and then ground right over there. Okay. All right. And then where you want to connect the pin for the breadboard is really up to you. I'm just going to pick any on the opposite side, and just make sure that they're not connected like that. They're like together on the same row, because that would just make it so that they're connected. You don't want that. So I'm just going to put them anywhere, just like that. Okay. So I have the sensor right now connected to my plant leaf, as you can see here. And I'm going to attach it to the breadboard. So right now the white one, that one's connected to the reference electrode, and then the blue one is to the sensitive electrode. So white is going to go to ground, and then blue is going to go to the analog pin. So let's connect that. So we've got white to ground and blue to the analog. There we go. All right, so that's the setup. I'm going to connect the battery pack later on so you can see that it can be used as a standalone. But for now, we're going to actually plug this into the computer, and then I can upload the code. So go and plugged in. Okay. And before I get into the coding again, do just want to mention that like the memory card is very easy to take out and put back in. I'm just using the 32 gigabyte memory card. So you just slide that in and then push it in, and it's good to go. Okay. So would I be able to return back to the code so I can show everyone how to upload it? I'm going to turn this volume way down. Hello. Okay. So let me switch back over. Okay. So I'm going to share my code again. So are you able to pull that up on the screen? I think right now it's still being shared.
Hey, Jenny, can you use your
Sorry.
yeah? Can you use your headphone to talk?
Yes. Yeah. Sorry. Yes. Okay. Were you guys able to hear me before, or was it a little bit hard to hear?
We could hear, but there is an echo around.
Oh yeah. Sorry about that. Yeah.
No,
like
so
two things, so it's a little bit of feedback happening.
no, now it's perfect. Don't worry.
Okay. Alrighty. Okay. So can you pull up the screen I have shared right? Yes, that's it. Okay. Perfect. Okay. So everyone can see my screen here, and I'm just going to pull out my sketchbook. So once it's been connected, you should see that your Arduino has appeared. For some reason, it's not letting me drop down in this menu. Okay, so let me just see if I can verify. It's not letting me click. Sorry, give me 2 seconds. I might have to stop sharing for a moment. Okay. So I'm going to try to reshare it, and hopefully it will let me click. Okay. All right. So now I'm able to access the dropdown. Okay. So if your Arduino has been connected properly, you'll see here that it's showing up as the Arduino in the port, and you'll have a nice little check mark there. If you do have an error, you're going to see just a red X, and it's going to be like, okay, well, your Arduino is not connected. So at the moment, everything has been connected. The light on my Arduino is green, it's on, it's working. And so I'm basically ready to upload my code. So all you have to do is basically say upload and save. And ideally, if everything works smoothly, then it should begin to upload the code, and it should come out saying success. So right now it's just taking its time uploading. Okay, there you go. So now it says success. It's been saved and it's now uploaded. So the code is now on the Arduino. And if we actually go to the serial monitor, we can see that the SD card is now ready to use, and it's starting to print some voltage readings. Yeah, there you go. And so in the code, it should be able to save everything to the SD card. So we're going to give it like maybe a little bit of time to kind of like upload. And what I'm going to do now actually is I'm going to just disconnect. But first I'm going to plug the battery in. So I'm just going to put it into the port. That way it can basically be standalone. Okay, so battery is now plugged in, and I can actually just disconnect this part, and it should still be on. All right, still on, so that's good. Not sure if you guys can see that carefully, that the green light is there and it's on. Okay. So, gonna let it run for just a little bit, and okay. So when you power off the Arduino, the memory stick will no longer record anything. So just as an example, if somehow you had some kind of like power outage, you know, obviously it's off, so the memory stick will no longer be reading data. But let's take it out and see if we can see what's on the actual card. So I'm just going to pull that out. And I have a handy adapter here, and that's going to go to my computer. And basically, I can just stick that in and plug that into my computer. Okay. So I've now pulled up the voltage readings that I got, and I'm going to try to see if I can share that with you guys. Are you able to, okay, there we go, perfect. So these were basically the voltage readings that we just got from going into the Arduino, and you can see we let it run for basically about 1 minute and 38 seconds. And so these are all the voltage readings that it's picking up. I do have another example where I've captured readings for 2 hours. So I'm going to share that with you as well, just so you can see. Let me just open that up. Okay. Okay, so on my screen now, I also have readings that I took over a 2-hour period. And you can see it's very easy to collect a lot of data. And you can basically let it run for as long as you have power. There you go. And another thing I do want to mention is that the file sizes are very, very small. The one where I'm taking readings for 2 hours is only 242 kilobytes in size, and the SD card can hold up to 32 gigabytes. So each data point does not use up a lot of space. And so you can store a lot of data just on a single SD card. Okay,
Jenny,
so
I have a question.
Yeah.
When you have more than one sensor, for example, 6 sensors, each one is going to create a new folder or a new text file?
No, so you can actually have it print to the same text file if you want. You can, if you want, print multiple text files. What I like to do, and I think it also saves a bit of space as well, is really just adding a little bit of additional code. So let me actually share with you, go back to the Arduino editor. So let me pull that up. Okay. So I'm back in my Arduino editor, and I do have where I've written for a case where you have 6 sensors. So this is slightly adjusted to allow for 6 sensors to be used at once. So now I'm using all of my pins. And if I scroll down a little bit, you're seeing here that it's able to collect all 6. And basically, you can just replace these values with, or add these into the demo file that I provided for you guys. So it'll print everything.
Great, thank you.
Yeah, no problem. It's all in the code, so yeah. Do we have any other questions regarding the demo?
Not so far. No.
Okay, so if not, I guess I can continue on with my presentation then. So let me go back to my PowerPoint. Okay, here we go. Okay. And I'm just going to scroll back down to where I was. Okay, that was a demo. All right, so I'm very happy to be able to share some of the results I got from using the sensors. So as I mentioned before, the sensors we received from the BEST group from the University of Glasgow, and they sent us 3 different types of sensors. I wonder if I can actually show you some examples of what they sent us. Okay, now we have a fabric sensor here. So here I have an example of a fabric sensor that they sent us, and here's an example of this silver chloride. And the one before was the carbon sensor, which you'll remember from a previous slide. So what I wanted to know was what would the voltage readings be when I put them in different pH solutions. So the pH solutions I used were buffer solutions with known pHs, and there was a pH 4 solution, a pH 7 solution, a pH 10 solution. And so from there, I was able to determine what voltage readings I would get. So what I found was that the carbon sensors actually had the greatest sensitivity compared to the other ones. We can see here that these silver chloride sensors did not perform very well. All the readings were quite low, and it was harder to distinguish between different pH solutions. So from there, I proceeded to only use the carbon sensors as they gave us the greatest sensitivity. So the next thing I wanted to look at was the hysteresis. And hysteresis is basically a memory effect. And so how you can test it is I started off with a low pH solution. I gradually increased the pH by adding a base, and then afterwards I brought the pH back down by adding acid. What we can see from the results here is that the first carbon sensor has some evidence for hysteresis. We are noticing that for around a pH of 4, once we increase the acidity, increase the basicity and then bring it back down, we are noticing that for a pH 4 solution, it's higher than what it was initially. So for carbon sensor 1, there's some evidence for hysteresis. Carbon sensor 3, on the other hand, we've observed much less effect of hysteresis, and we can see that when we increase the pH and then bring it back down, the voltage readings are low, just like they were in the beginning. And carbon sensor 2, this was slightly transformed to allow for consistency between the 3 sensors. And the third result I wanted to share was some readings I took from live plant tissue. And so I wanted to see whether or not there's a difference between healthy plants and white mold infected plants. And so I took a ton of readings. I do want to rerun this experiment again because I wasn't as confident with my setup as I am now. So I do plan to rerun this one. But despite these being fairly preliminary results, the results do suggest that the infected plants have a slightly lower pH than the healthy plants. So what can we conclude from all this? The preliminary results really indicate that these miniaturized pH sensors have the potential to be used for high throughput phenotyping and disease monitoring in plants. And the use of an Arduino platform in conjunction with these miniaturized sensors has been confirmed to be a cheap and reliable way to collect large amounts of data over a long period of time, assuming there is a continuous power supply. Where we want to go next with these is, well, the development of these sensors for use in plant tissues is still in its infancy, and so more studies are required for testing and validation. As well, we need more validation under a number of field conditions for white mold infection, and eventually we want to transition this to the field. As well, additional power solutions are needed for the Arduino to be standalone for extended periods of time. At the moment, these 9 volts battery can last up to 2 hours, but if we want it to be collecting data for days or even weeks, we will need a longer term power solution. That being said, if you do have a continuous power supply, then you don't need to worry about this at all. I think it's mainly for if you're going to be bringing this out into the field where you may not have access to like a power cord or something. You will probably need to have a long-term power solution. Another thing to note is that the large quantities of data that ends up being produced by the sensors need to be processed in a very efficient manner. And so all in all, our lab is still in the process of developing wireless, disposable, and compostable sensors that can be readily implemented in the field. All right, so on to my acknowledgements. I'd really like to thank my supervisor, Dr. Villar Hoyos Viegas, as well as my committee member, Dr. Philippe Seguin. I'd also like to thank all of my lab members for their continuous kindness and support. And I especially like to thank Seamus, who helped me extend the length of these sensors, because when they sent them to us, they're basically very short. So thank you so much, Seamus. And let's not forget our collaborators. Thank you so much, Dr. Ravinder Dahiya and Dr. Libu Manchakal. And of course, Fabienne Frankel. Thank you so much for all your help. Thank you so much for the sensors. And thank you guys for listening. Here are some links, and I believe they're already in the comments, so you guys can go access them. Here are my references if anyone's interested in some further reading. And I'd like to take some questions now. So thank you so much.
Yeah, great, Jenny. You did a really amazing job.
Thank you. Thank you.
It was really easy to understand, and I feel excited to start trying myself, you know, making...
Yeah, for sure. I'm like, if I can do it, you can do it. And it was such a pleasure to share all my work, my research with you guys. So thank you so much for coming.
Well, I have a question, but well, it's like, say, like, so I don't know, can you, so like we have seen Raspberry Pi and Arduino, so what are the advantage and disadvantage of Arduino compared to Raspberry Pi? Like, are they similar compared in terms of, like, can someone do what you are doing also with the Raspberry Pi, or like, can you say what are, like, for you the advantages or disadvantages?
Yeah, so that's a great question. I'm really glad you asked. So I have worked with Raspberry Pis. I am, well, I was also in the process of trying to use Raspberry Pi as a automated camera so that it can take photos. What I have noticed with the Raspberry Pi is it does use less power than the Arduino. So the Arduino is slightly more power extensive. It drains more power. So that's something that people may need to consider if they are going to be implementing this long-term out in the field. As for whether or not this experiment can be conducted using a Raspberry Pi, I haven't actually tried that myself, so I wouldn't really be able to give you a definitive answer. But I mean, I don't see why not. I think it is definitely possible, and if you put your mind to it, you can most likely come up with some kind of solution.
So you are saying that Arduino will be more like suitable for like phenotyping in, like, not outside in the field, but more like in
a lab environment or a green
more stable there.
Yes.
That's cool. Oh, we have a question. Very interesting. The pH sensor is commercial in the lab?
Yeah, so these sensors, because we received from the best group. I think they developed them. So we didn't make them ourselves. They sent them to us. So I don't know whether or not they plan to make them commercial or not. I think it's up to them. We were just testing them out and using Arduinos to see if they work. So you'd have to contact them.
Yeah, great. Dr. Valeri just post that in making collaboration.
Yeah.
Cool. So thinking about application on field in the future, for example, that it is important to make a container for leave these outside. Right?
Yeah.
And we have the weather and other conditions.
For
you see how far from now you think that, for example, breeders can have access to this technology to be taking these measurements in field?
Yeah, so at the moment, I do still think that this is fairly preliminary. The end goal would be to have them out there standalone, but there's definitely some hoops to jump through when getting to that point. Right now, as I mentioned with the Raspberry Pi that I've been trying out, I was able to build a little container for it to be in, and it was able to withstand weather and still be on. So this, if you wanted to just try that out and just have a little container for the Arduino to be in, I think you could probably do that. I don't see any major barriers, as long as you make sure that it's sealed so that water can't get in, and you are able to ensure a long-term power solution. I think that's where the barrier really is, making sure that it can run continuously, because the moment it loses power, you're going to lose your data. So again, just having that continuous power supply, but in terms of weatherproofing, I don't think it would be too hard to do.
Yeah, great. I have another question. Imagine a student that is just starting working with this. Do you recommend do some specific class to learn about electronic, to learn about this technology, or you think following tutorials and pipelines, the student can make their own platform like you did? Did you have classes, because you really know about that? You know?
I know. So I learned everything just online. I did get a little bit of help from our collaborators in Glasgow. They recommended some links and websites for me to look into. But yeah, everything I was able to find online. I personally don't have a background in electronics. My background is actually molecular biology and genetics. And I'm more knowledgeable of plants than I am about anything electronics. So I did learn all this on my own, just with the help of the internet. And the Arduino website itself has so many resources that people can access, and a lot of people publish projects, and all of it's open source, so anyone can just go and pick one up and start building their own creations.
That's so great.
Yeah,
I mean, like, thank you for telling us what is your background, because, you know, like, I think Phenomics brings so many people from different backgrounds, and it's very cool that you can always like change or learn.
Yeah, for sure. Yeah, I think integrated science, really important. Just making sure that, you know, we're making all these different connections, and our collaborators have been so helpful to us as well. So
we have another question. I don't know if you can answer. I'm
okay.
gonna say it like, hi, I'm in India. How can I get the sensor if I want to test it in our lab?
Yeah, so please contact the BEST group. If you let them know whether you're in academia, let them know and reach out to them, and they might be able to send you some.
Thinking about other applications for this sensor, right now you are saying how the disease can affect the pH in your samples. Do you have any other application or suggestion, or possible other sensors that a person can find online, like pH sensors that someone can use in application?
Yeah, so I think in the slides, I did mention a number of different methods for measuring pH. So this one is definitely something that not many people have looked into specifically for measuring pH in plant tissue. The sensors initially were designed to be used for medical purposes. They're like wearable sensors, especially these fabric ones. Basically, the idea is to place them onto your I guess arm, and then you can measure the pH of your sweat, and that can be used for medical purposes. In terms of plants, these have not been tested out in plants at all, and that's what our group tried to do. So I think the best group, they really are on top of the game in terms of designing these sensors and testing out different types of materials, and they were able to test out some different materials for us as well. So for other sensors, I'm not sure of any other groups who are developing these types, but perhaps you can look in the literature and see what's out there.
There is another question.
Mm-hmm.
Is there a possibility to do the same job with another sensor, for example, temperature and humidity sensors?
Yeah, for sure. There definitely, you can definitely do that. The Arduino, there's definitely code out there where you can have it take temperature readings. And I think with temperature sensors, these I would say are much more, I think, stable, reliable, and much more developed than pH sensors. Same with humidity. So for sure, you can apply the Arduino to taking these types of measurements. Again, you just have to find the code and the setup. So.
Yeah, great. Are you working, breeding population, or is the same genotypes with different treatments?
Yeah, so when I want to run my experiment again, we do have some plants that have moderate white mold resistance, so I am planning to test those out. They're called white mold 1 and white mold 12, and they were sent to us. They are pintos. And I think right now I'm just going to stick to working with the same market class with pinto beans. So I want to see, high and low or moderate resistance white mold resistance plants, and see whether or not I can find out differences in terms of pH between them.
Great. Yeah, interesting.
Thank you.
Anna?
Yeah, thank you so much. It was very great. Yeah, so thank you for doing this great workshop for us.
Yeah, no problem.
And if you also, like, as we say to all the speakers, like, please come back if you have any other evolution about your setups.
For sure. Thank you so much for having me. I had so much fun sharing all my work and doing the demo for you guys. So yeah.
Yeah, that's
people
great.
are saying congratulations and thank you.
Yeah.
Of course, like, I don't know if you are part of the Slack channel, but if you guys have any question for Jenny, you can also reach us and contact her directly.
Yeah, I can also include my email just in the chat, and if anybody has any questions, they can also email me, and I will try to answer them as best I can.
Yeah, that's great. Yeah, I was thinking you should have your YouTube channel too.
Yeah,
you're great. You're great. Yes.
Thank you.
So thank you for coming, and let me know when you have new high tech or tech or pipeline to share with people here in our community. Okay.
Sounds good.
All right.
Thank you so much.
Let's go on. Let me share here.
Yeah, so as I anticipated last week, we have a new workshop, which will be the last for this season. Then we will take a break for the summer. Actually, yeah, so we will have Paulo Flores and Diego Gris, that they will be talking about the basics of drone flights and data extraction for small plot research. They are from North Dakota State University. So if you register, this is very interesting because it's the we have been not so many workshops about drones, and people are like insisting. So that's very great. So thank you, Felipe, for suggesting them. So if you register to this workshop, you don't need to register again. We will send you all the information on the Thursday, as we usually do. But please share this link with anyone that can be interested.
Yeah.
And yeah, thank you for being here today to listen us, and have a nice weekend.
Yeah. See you guys. Thank you for coming. And next Friday we are here again. Bye-bye.
Bye-bye.